Okay, so I want to start with the disclaimers that I'm not a population geneticist. I know, but prior to this meeting, I knew absolutely nothing about cancer and mathematics of cancer. I'm talking mostly about analysis. So the work I'm going to talk about here was done jointly. The analytical work was done with Rafe Mazzeo, and then the numerical work is actually not done so much jointly as with Jon Wilkening, as it's almost entirely the work of Jon Wilkening. Okay, so we've had people have alluded to the existence of this subject, population genetics, and so I'll describe a little bit more what it is. So we study how the distribution of variance in a population, the reproducing population, evolves over time. Typically, there are four important events. So the randomness in the number of offspring of a given individual or pair in a given generation is sort of the random element in population genetics. Mutation from one type to another, differences in the fitness among the different types, and possibly one can talk about migration in and out of a given environment, which we saw a lot of in the previous talk. Now, the simple model, which is the only model I'll talk about, and it's one of the most important models, is the so-called Wright-Fisher model, where you assume that the population size is fixed. So in the simplest case, you just assume that you have two types, an ancestral type, capital A, and a derived type, little a. So these are alleles. And we assume, first of all, to start out with, that they have the same fitness and that there isn't any mutation. So since the sum of the number of little a and the number of big A is fixed, you just have to keep track of one of them. So we'll keep track of the number of little a's, which we think of as the mutant type. And the model is just a Markov chain. And all we need to tell you then is if we know that we have I of type little a at time t, what's the probability of having j at time t plus 1? And in this setting, we use the random mating model, which is just the simplest possible model of random mating, where you have an urn with colored balls, and you just pick out balls and with replacement to create the next generation.
I think there is no mating here, actually. This is
there's no real mating. We use that term. We use that term rather loosely. I mean, there could be mating, but we're not going to discuss it. Okay, so this means that the probability is just given by this binomial distribution here. Okay, so now suppose we'd like to incorporate some other effects. We'd like to incorporate mutation and selection. And the way we do this is we just change the odds. So back here, we just have the odds of one type versus another type. And what we do is we just change those odds to allow for selection. So we'll say type A has, a little type A has a selective advantage or a selective difference. So its fitness is 1 plus S as compared to 1 for type B, big A. And there's a rate of mutation from little a to big A, which is mu1, and a rate of mutation from big A to little a, which is mu2. And then if we change the probability of mutation, of getting a type little a to that, slightly complicated looking formula, then the transition matrix just becomes the same matrix, same binomial sampling, but with sort of a different coin. It's coin flipping, but with a different coin. Okay, so there are lots of generalizations. I can include more types. I could have multiple populations. I could have migration. But I'm only going to talk today about the simplest case, because it so happens that from the perspective of numerical analysis, even the simplest case has never really been adequately dealt with in the literature. Okay, so as is not uncommon in this field, people would like to replace a discrete model with a continuous model, so they obtain what's called a diffusion limit. So what you do is you rescale the population so that the population now is a frequency, takes values between 0 and 1, and then you change the time. So the time is now in multiples of the population, Tn. And this is a continuous time stochastic process. You can take a limit as n goes to infinity, and you get a process on the interval 0, 1. So it's a random walk on the interval 0, 1. And these things are described by partial differential equations. And this equation here is the backward Kolmogorov equation. Okay, so it has this second, I'll talk about the individual terms in a minute. So first of all, these coefficients, the selection term is this term here, and its coefficient is defined as the population size times the selective advantage or disadvantage. And similarly, the mutation terms are defined by multiplying by the population size and using the mutation rates. Now, we assume that those numbers are fixed as n goes to infinity. And a lot of confusion when people start to hear about this is they say, well, does that really mean the selection coefficient changes as the population size increases or the mutation rates change? No, the answer to that is no, they don't change. They're really physical parameters that you measure them. What it means is that when you use the model with a discrete sized population, you have to scale the coefficients properly. That is, you have to pick a size for this number n. Because what you really do is you solve the problems using the diffusion model, but then you plug in the discrete values for a particular size of n, and that tells you how to pick your sigma, your M1, and your M2 from known mutation rates and known selective advantages. So there is no assertion here that these kind of very physical parameters actually change with the population size. They certainly don't. Okay, so that was the backward operator. The formal adjoint is the forward operator, and I call these Kimura diffusion operators. Now, as to whether or not Kimura was not the first one to write them down. Probably they were written down in the 30s. But Kimura was the first one to really use them and spent the 50s and 60s proving an incredible number of things in population genetics using this diffusion approximation. Okay, so if we denote the fundamental solution for the diffusion equation. So it solves both equations. So the dt of this function, p sub t, is on the one hand, it's the forward operator and the backward operator in the x variables, or on the other hand, it's the forward operator in the y variables, because they're formal adjoints. And the initial condition is that we get the identity as t goes to 0. And how do we understand this? Well, if we start out with an allele at time 0, which is at frequency y, then the meaning of this function is just that the probability that at time t, the frequency will be between a and b, you just get this by integrating from in the x variable from a to b. So that's a sort of a continuous Markov chain. Continuous time, continuous space Markov process. Okay, now in the limit, the second order term, x times 1 minus x over 2 d by dx squared, this is related to the randomness in the number of offspring. Now, strangely, and this is a little peculiar, the mutation and selection, so most people think of mutation and selection as being random processes. However, in this model, relatively speaking, mutation and selection become deterministic forces because they're represented by a vector field. So if you think about what that means, that if we didn't have this diffusion term, this x1 minus x d by dx squared, and we just solve the equation dtx is given by this vector field, it would just give us a single trajectory. It would be deterministic. It would be a deterministic model. So in this diffusion limit, the interesting thing that happens is that, relatively speaking, mutation and selection are deterministic relative to the randomness of mating. Okay, what makes it difficult to study this operator, both from the perspective of analysis and from the perspective of numerical analysis, is the fact that the coefficient of the leading order term vanishes at the boundary. Okay, so starting out with the process with no the paths, so you can think about having a stochastic process where you have paths. The paths of the process satisfy this SDE. So dx, the differential in the path, is square root of 2x, 1 minus x times dw, which is some kind of white noise process, plus the vector field b of x times dt. So the drift is b of x dt. So I should say, I learned about this subject in a DARPA project, the so-called Fun Bio project, and it only took us a year to realize that when the biologists talked about drift, they meant something exactly opposite of what the mathematicians meant when they talked about drift. After that, things went a lot better. Okay, so now, the fact that the coefficient of the second order term vanishes means that if the vector field vanishes at a boundary, b of 0 is 0 or b of 1 is 0, then when a path of the process would reach that boundary point, it would stay there. It would not come back into the domain, and it would not go any further. It would just stay there. So 0 and 1 can be absorbing states, and whether they're absorbing states or not just depends upon whether the vector field is 0 or non-zero there. Yes?
So, I understand that you call mutation selection deterministic because they only enter in the B of X and not in the first term that describes
Yes, right, because they only appear in the vector field.
Yeah, but that is arising from the assumption that you have weak selection in the diffusion approximation.
Yeah, I'm just saying relatively speaking. They're deterministic. Relative to the random mating, they're deterministic. They're not deterministic in real biology, of course.
You confuse me because you always talk about mating. To me, this is just a haploid, whatever. Process.
Yeah, I'm sorry. I told you, my understanding of biology is very limited.
Okay, but what exactly do you call mating?
Oh, this is just going from generation 1 to generation 2. It's just paths of the process.
Okay, good, thanks.
It's just that. It's just this process of selecting colored balls out of the urn.
Okay.
I have a question. So with mutations, why are they absorbing states?
Oh, if there are mutations, they're not.
Okay, fine.
So I'm saying,
in a general problem, they're not absorbing states.
yeah, no, well, if B of 0 is 0, then 0 can be an absorbing state.
Okay.
If B of 1 is 0, then 1 can be an
In
absorbing
general, if
state.
you have both mutations,
Then they're no absorbing states, and instead you have a stationary distribution. Okay, so applications. So the basic application of this forward model is to understand how distributions of variance in an ensemble of populations evolves over time. So the forward equation actually acts on Borel measures, and in fact, this is a complicated analytic issue because, well, it's this technical fact that continuous functions is not a reflexive Banach space. So you end up with this kind of strange functional analysis for the forward operator. So the forward operator generates what we call a semigroup, which is just the propagation of the population. But it's only strongly continuous, that is, it only really has nice limiting behavior as time goes to 0 if the measures are absolutely continuous, that is represented as a h of x dx times, where h of x is an integrable function, and the possibility that you have delta measures at 0 and 1 if the vector fields happen to vanish there. Okay, so then the solution of the equation dtw is this LTW describes how the mass of this measure is redistributed. If alpha times beta is not 0, if both mutation rates are non-zero, then there is a unique stationary measure, which is the null space of this vector of this second order operator. And what happens is all of the solutions to this PDE will converge to that measure as time goes to infinity, or some multiple of that measure. If one of the alpha or beta vanishes, then the null space of the forward operator is a delta function at that boundary point. So it's delta of x dx or delta of 1 minus x dx. And if both vanish, then both delta measures are in the null space. So this is just another way of saying that in that case you have either 1 or 2 absorbing states.
Sorry, by now I'm lost with the notations. Remind me, alpha and beta were in the original.
Yes, alpha and beta, I think, is unfortunately something I didn't define yet. Those are just the scaled mutation rates.
Oh, okay. I see. Okay, then it's obvious.
I'll
Okay.
define those in a second. I'm
Okay,
sorry.
Okay.
Okay, so the simplest application was the application I just described, but that application isn't really used very much in actual applications. The actual application is to this so-called allele frequency spectrum, which is more or less the same thing as the site frequency spectrum, which is closely related to what the previous speaker was talking about. And it has to do, and the reason that this is of interest is this is something that you can actually measure when you do sequencing of cells. You can actually measure the slight frequency spectrum. So what you imagine here is that you've sequenced N chromosomes or N genes from a much larger population, and what you find are that there are K sites on these N chromosomes where there's some variation. So again, we assume the same thing that was assumed before, the so-called infinite sites model, that either a site is mutated, unmutated, or there's exactly 2 alleles. So you don't have multiple mutations at a given site. And there are no back mutations either. So the allele frequency spectrum is then the sequence of numbers F1, F2, Fn minus 1. So it's given by the number of chromosomes that we've sequenced, and each Fj is the number of sites where an allele appears in J samples. So we're not actually keeping track of the different sites. We're just keeping track of how frequently an allele appears. And it turns out that in the large N limit of very, very many samples, these frequencies actually satisfy a Kimura diffusion equation. The statistics of these things are affected by just about everything. Changes in the population size, mutation rate, selection, just about everything. And when you make these observations, what you want to do is you want to have some kind of a parameterized model, and you would like to say, well, what is the likelihood of these observations given this choice of parameters? And so then you have to have some way to compute those probabilities. And there are basically two ways to do this. And one way is to simulate using the Kimura diffusion equation, and the other way is to use these coalescent trees, so essentially these lineages from the previous talk. So here's an example, just because I find this to be an incredibly difficult thing to think about, the allele frequency spectrum. But here we have 6 samples. We've found 8 sites that are variable. We're just thinking about having single nucleotide polymorphisms. And as we see, there's 1 sample with that guy, 2 samples with that guy, 3 samples with that guy, so on and so forth. And then from these 6 samples, there are 8 segregating sites, 8 places where there's variability. And then the allele frequency spectrum is 4 sites where there's appearing once, 2 times where it's appearing twice, 1 time where it appears 3 times, no time where it appears 4 times, and 1 site where it appears 5 times, right there. Okay, so you typically expect many, many, many more things to occur at low frequency. And in fact, in the infinite sites model, there's an infinite number of members of the population with no mutations. Okay. So a lot of problems in population genetics have been addressed using these diffusion models. These Kimura, Crow, Ota, Owens, more recently Patterson, Evans, Schvetz, and so forth. And I got interested in this because I heard a lecture about 10 or 15 years ago about the percentage of human genetic material that comes from Neanderthals. And part of the calculation of this percentage has this inference loop in it, and the sort of inner loop of the inference loop was computing the evolution of the allele frequency spectrum. And Steve Evans said to me, and he did a lot of this work, he said, we couldn't solve the diffusion problem accurately enough, and so I simulated the stochastic process. And I'm thinking, boy, that's insane. You simulated the stochastic process, so you get convergence at the rate of 1 over the square root of n, where n is the number of paths. So that's very, very, very slow convergence. You don't get a lot of digits in any finite amount of time. So I thought, boy, that's sad. We really should figure out how to do this. And so I asked Jon Wilkening, and I have to say, I think my greatest contribution to this field will turn out to be that I asked Jon Wilkening to work on this problem. Because as far as I know, he's the only numerical analyst who could actually have done what he did. Okay, so let's talk about why this is hard. It's hard because of the boundary conditions. And the boundary conditions are complicated because this coefficient vanishes. So this problem was addressed in a very remarkable paper of Feller in 1952, where he said he started out with an operator of this general type, and he found all possible ways to take this formal operator, which is an unbounded operator on continuous functions, and define a semigroup which was continuous and positivity preserving. It's a remarkable piece of work. And we're going to only talk about the simplest possible boundary conditions that you can apply and get a good functional analysis for you. Okay, so
You said continuous, you didn't mean the operator continuous.
What's that?
You used the word continuous, you didn't mean continuous, it's only closed. Okay.
it's continuous. It's a closed operator on continuous
Right,
functions.
right,
An
right,
unbounded
right.
closed
Right, yeah,
operator,
yeah.
trying to avoid technicalities.
Sorry, what is C of X in the equation?
C of X is just you can have a 0th order term. Okay, it's like, in these models, it's usually like a killing term. It's called a killing term. So you kill off members of the population. I'm not going to talk about it. It's just something that was allowed in Feller's work. We don't see it very much in population genetics, but it can be there. Okay, so the simplest choice for getting a closed operator is what's called the graph closure. So you just take the operator, you let it act on C2 functions, and then you define this topology where you just take the C0 norm of the function plus the C0 norm of the operator applied to the function. You just define the space as the closure of C2 with respect to this topology. And I denote the operator by L bar. And it defines a strongly continuous positivity preserving semigroup. And it's probably the most useful one in applications. The domain of this operator is a subset of C0 functions intersect with C2 functions in the open interval, but complicated things happen at the boundary. And what happens at the boundary really depends upon alpha and beta, which have I still not defined them? No, I defined them. Did I define them in the previous slide? Yeah, there are alpha and beta. So alpha is the mutation rate to 0, and beta is the mutation rate to 1. Okay, so what happens really depends very strongly on alpha and beta. So if alpha is positive or beta is positive, then a function is in the domain provided that the first derivative has a limit at that boundary point. So if alpha is not 0, the first derivative has to have a derivative at 0. If beta is not 0, the first derivative has to have a limit at 1. Now, we call the solution of the diffusion equation from this particular choice of closed operator the regular solution. In population genetics literature, this is often called the no-flux solution, because it can also be defined by a condition that looks like a flux condition on the first derivatives at the boundary, scaled by certain factors. Now, the key thing to understand about the regular solution, and this is what makes it very strange and very different from ordinary non-degenerate PDEs, is that the solution to the equation is uniquely determined by its regularity at the boundary, given the initial data. It's determined by its regularity. That is to say, you cannot specify its values at the boundary. You cannot specify its normal derivatives at the boundary. It's the regularity at the boundary that makes the solution unique. And this is what makes it very hard to approximate this numerically, because you cannot impose a regularity condition using a finite difference approximation. You've taken a finite number of samples. How the hell do the samples know whether they're samples from a C0 function, a C1 function?
Well, but you can multiply by the thing that has the right regularity and then solve it using finite difference.
Sometimes.
Okay, well, that's how physicists do that.
Yeah.
I just, you know.
But it's very hard to do it accurately. It's very hard to get digits that way, let me tell you. Okay, so the regular solution also has some remarkable properties. It has the following amazing property, that if you start with data that is Cm, it has m derivatives up to the boundary, then actually the regular solution has m derivatives uniformly in time up to the boundary, which is a very strange and unusual property, in addition to being infinitely differentiable as soon as the time is positive. Okay, so there's another boundary condition that you can specify as long as the mutation at the boundary effect is not too strong. So here I've scaled everything. So the coefficient of the diffusive term is 1. It's x1 minus x d by dx squared. So if alpha and beta are less than 1, then you can also define a boundary condition using the Dirichlet condition. That is, you can insist that the data goes to 0 at the boundaries. Now, if you do this, and alpha is not 0, then the solution at 0 will automatically have that kind of a singularity. It will behave like x to the 1 minus alpha. And similarly, at 1, it will behave like 1 minus x to the 1 minus beta. So again, it can't be the same solution, because I said the solution which is regular at the boundary is unique. So what happens is, if I specify 0 boundary conditions, then I end up with singularities at the boundary. So again, from the perspective of numerical analysis, it's a nightmare to deal with singularities at the boundary. If alpha happens to be 0, then you get an x log x singularity at one end, and if beta is 0, you get a 1 minus x log 1 minus x. Okay, so, if we want to impose an inhomogeneous Dirichlet condition, that is, we want to set u equal to something, u of 0 and t equal to something, and u of 1 and t equal to something, that what we would do is we'd write the solution like that. We'd put in this linear term. The linear term is annihilated by the drift term, the second order term. And what happens is then u0 becomes a solution of the regular, a solution with homogeneous Dirichlet conditions, which means that u0 is going to be singular. Okay, and the problem that we're most interested, you have to do this. Okay, so the motivating example comes from a paper of Evan, Schvetz, and Slatkin, where they're studying the allele frequency spectrum, and they want to use the Kimura diffusion to estimate quantities connected with it in a varying population. So here I said when I constructed the model, I said we assume the population is constant but goes to infinity. Well, for other reasons, you can allow now the population to vary, and what you do is you just change the coefficient of the diffusion term. So you put 1 over the population size there. Okay, and they want to solve this equation. They want to solve dtu is 1 over 2 times the population, x1 minus x dx squared, minus the selection term x1 minus x dxu. And they have an initial condition, and they have a non-homogeneous Dirichlet condition. So they want to have u of x and t be sigma theta, which is some parameter which is determined by a mutation rate, times the population. Yeah, there aren't. But this is for the allele frequency spectrum. It's not. So the only way that the mutations appear in this equation is we're injecting them at the boundary. We're just assuming that mutations are arising at 0 frequency at the boundary. So at very, very low frequency at the boundary. So you get this theta rho of t. And in fact, the allele frequency spectrum is not u. It's u divided by x times 1 minus x. And this is, so you might say, well, that's a little peculiar. It looks like it goes to infinity as x goes to 0. And indeed, it goes to infinity as x goes to 0. And that's, again, what I was saying. Sort of in this scaled limit of an infinite population size, you're going to have infinitely many alleles that are not, you're going to have infinitely many sites that are not mutated at all. And so that's why this is fine. But fortunately, when Evans, Schvetz, and Slatkin derived the equation, you see this denominator disappeared. And you end up with the perfectly nice PDE for the numerator of this term. So we're going to actually just study the numerator of the term. OK, so this is a very hard problem for a whole range of reasons. The population size is changing. You're injecting data at 0 times 0, which means that you have solutions that are singular, inevitably. And it turns out, it so happens that the initial data, which comes out of sort of an equilibrium distribution, assuming the population size was constant for a very, very long time, is inconsistent with the boundary condition at that corner point, 0, 0. So all possible things that could go wrong in this problem. OK. How long do I have?
Well, I don't know. I wasn't really keeping track, but we have 40 minutes altogether, so like 15 to like 20 minutes.
OK, I just don't remember when I started. OK. Maybe I just skipped that. OK, numerical methods. OK, for many, many years, people have been solving the Kimura diffusion equation numerically. In particular, Kimura solved it himself. And as long as you're looking at just this piece here, x1 minus x, with these two linear mutation terms, you look at that and you say to yourself, huh, those operators map polynomials to polynomials for every degree. And then you say to yourself, the eigenfunctions of that operator must be polynomials, because those are finite dimensional spaces. So we're just talking about finite dimensional linear algebra. And in fact, they are. And the polynomials are classical, what are called Jacobi polynomials. So this is the eigenvalue equation. Those are the eigenvalues, minus n alpha plus beta plus n minus 1 times the Pn alpha beta. And Pn alpha beta is some constant times this classical Jacobi polynomial, Pn alpha minus 1 beta minus 1, evaluated at 1 minus 2x. So the Jacobi polynomials are normalized usually to be defined on minus 1, 1. And we're working on 0, 1. So that's how you have to scale the variables. In cases where alpha and beta vanish, you have to take limits, and you get similar kinds of results. But what happens is at the endpoint where alpha or beta is 0, the eigenfunctions will vanish there. So you get an x or a 1 minus x multiplying the eigenfunction. All right, so the solution to the PDE is given by an infinite sum. These are the initial conditions determine the coefficients. I represent my initial function in terms of these eigenvectors. And then this is the formula. Now, you might say, oh, well, I guess there's nothing more to say about this. But unfortunately, the problem is that it's actually very difficult to compute the polynomials correctly. And I think that this is a problem that arose in a lot of biology papers, is that they sort of tried to use a naive approach to computing the polynomials. That is to say, they wrote down the polynomial as the sum of coefficients times x to the n. And then they plugged in values of x. And the problem is, when you do that with these kinds of polynomials, the coefficients start to grow extremely fast. They grow exponentially fast. And then what you end up with is sums of terms of different signs that are extremely large. So when you do these calculations in floating point arithmetic, you run out of digits. And when you try to compute polynomials this way, you just get garbage after a fairly small number n. So what you actually do are you use these things called three-term recurrence relations. So I have the n plus first polynomial at the point x is some constant times the nth polynomial at the point x times this linear factor minus some constant times the n minus first polynomial at the point x. And these arise because these polynomials are orthogonal with respect to a certain weight. They're obtained by applying the Gram-Schmidt process to monomials with a certain weight factor, which comes out of the operator. And the coefficients there can be computed, and using these, you can actually compute these polynomials very quickly and very accurately. So again, my glorious collaborator produced this incredible slide. So he said, well, let's look at the 23rd polynomial with alpha is equal to 1/4 and beta is equal to 3/4. And what we're going to do here, so the black line is what we get if we compute it using a recurrence formula, the 3-term recurrence formula. The green is what we get if we just try to sum up the monomials. And what you see is, once you get around pretty close to the right endpoint, you just have absolutely nothing left in the way of digits. Now, I have to say, this is the last time you get anything. If you go to n equals 24, what happens is the graph for the green graph would go way off of the edge of the plot. So that's why we plot n equals 23. That's the end of the story for summing up the terms. This is just using this is called Horner's rule, which is considered to be a more stable way to compute polynomials, but also, as you can see, totally fails in this case. And what happens here, if you look at the coefficients, what you see here is when you reach this point, n equals 23, the coefficient, the largest coefficient has gotten to be about 10 to the 17. So there's just no digits left in a floating point machine.
But if you go back to the recurrence formula, you have this A's and B's or something, ANs and BNs.
Very moderate terms.
I see, they don't blow up.
No. Everything is very moderate. Because you see, in the end, these polynomials take values basically between -1 and 1. Nothing crazy happens with the actual functions. And that's why, again, you're summing up all of these different terms, but in the end, you're getting a number that's between -1 and 1. But if you're summing up terms that are 10 to the 17 minus 10 to the minus, you end up with no digits in floating point arithmetic. So you have to be careful. And I think that part of the reason people stopped using this method in population genetics is that they didn't know how to compute the polynomials accurately. So I just mentioned this because it's not something everybody knows. Numerical analysts know this, but people who aren't numerical analysts don't know this. Okay, so the eigenfunction approach is only directly applicable to the case of, I didn't realize I could do this. Okay, let's see. Of the operator L alpha beta with no selection term. If we want to include selection, or if we want to have an inhomogeneity here, we need to use some other method. And what I'm going to talk about for the next few minutes is other methods for doing it if you can't use eigenfunctions exactly. So here, let's suppose we have just a selection term, x1 minus x times s of x d by dx. It's, in a very precise mathematical sense, a lower order perturbation. It's what's called a relatively compact operator compared to L alpha beta. So it really is a small perturbation. It doesn't change the domain of the operator. Okay, so we're going to talk about two methods. The first employs orthogonal polynomials defined by this weight function, x to the alpha minus 1, y minus x to the beta minus 1, e to the s of x, where s of x is just the indefinite integral of little s of x. If s were 1, this would be the weight function with respect to which the Jacobi polynomials are orthogonal. So if s isn't 0, if there is selection, these functions are not eigenfunctions. But if you choose the sample points carefully, the discretization you obtain using this basis is what's called an exact Galerkin approximation. So what that means is I'm going to pick a subspace, a finite dimensional subspace of data, and I'm going to find the best solution I can to my equation restricted to that subspace. It's not an exact solution of the equation, except when I restrict it to that subspace. I project it into that subspace. And if you pick the sample points carefully using this basis, you get such an exact Galerkin approximation. Once again, you evaluate the polynomials using 3-term recurrences. I'm not going to describe this in detail. It's a pretty standard method, or fairly standard method. Our second approach is less standard, but really, in many ways, a better idea. So it starts with something known as Duhamel's formula. So Duhamel's formula is what you get if you multiply the equation by the fundamental solution for the L alpha beta part, and you just integrate by parts a bunch of times. You get the following formula. You get what looks like a formula for the answer. U of x and t is given by e to the t alpha beta, so the solution operator applied to the initial data, plus this integral from 0 to t of e to the t minus s, L alpha beta, applied to this term, which is sort of a perturbation term. So really, it isn't a formula for the answer. because, of course, the answer appears on both sides of the equation. So I can't just say, OK, I can compute the answer this way. But what it is it's an integral equation for the answer. And what it is, in effect, is it's in a sort of, when you discretize this equation, you're essentially discretizing the solution operator for the problem rather than the differential equation itself. And this is another fact that's not widely known outside of numerical analysis. When you discretize a PDE, you inevitably run into ill-conditioned problems. That is, the matrices you have to invert, their condition numbers get larger and larger, the finer the discretization becomes. So eventually you get to a point where you can't get any more digits. And the reason is, well, you want to make the mesh size smaller to get more resolution, but the problem is the condition number of the problem is proportional to 1 over the mesh size squared. Once that number gets to be bigger than, the number of digits you get is 16 minus 1, the log of that. And once that gets to be bigger than 16, you don't get any more digits. So on the other hand, if you work with the solution operator, it never becomes ill-conditioned. It's just a question of being able to compute the integrals accurately. OK, so the main challenge in this approach is finding a way to compute the kernel of e to the t alpha beta, the solution operator for the model problem. So the simplest approach is you just write down an eigenfunction expansion. You can do that. Certainly, that's fine. Where these are appropriately normalized eigenfunctions, it's a rapidly convergent series in principle, at least for t a little bigger than 0. But the problem is that the way that we'll use it, so remember, it appears here in this term. Here it appears as e to the t minus s L alpha beta. So t minus s is never going to be a big number, because we're just going to go one time step. So t is never going to be bigger than some little h, and t minus s is going to be smaller than that. So that's not a particularly good way to do it. Summing the series, you can't get too much accuracy doing that. OK, so what we do, now it's going to become,
5 minutes or so.
yeah, OK. The last 5 minutes will be totally incomprehensible anyway. So we're going to split our little interval, 0h, into subintervals. So we pick these C sub j's very carefully. They're what are called Gauss-Lobato quadrature nodes. So these are specially selected numbers that sort of accumulate near the boundaries of the interval. And then what we're going to do is we're going to try to find, we have our initial data to u0. We're going to try to find functions u1 through u nu, so that if we use polynomial interpolation in time to define the action of the perturbation term, the resulting samples of the function at those times are given by ur exactly. So this means we're going to replace our continuum model with a discrete model right here. U sub r is this e crh L alpha beta u naught plus this sum of terms, where this is the solution operator applied to the perturbation term. So e rj of z, that's just the solution operator, because I'm going to substitute in for z h times a. So this is really an operator acting on a vector. And we're going to just use the Lagrange interpolating polynomials. So that is, we're just going to be able to pick the values at these interpolation points, these so-called Gauss-Lobato nodes. OK, so we rewrite the system of equations. It looks like that. It's the identity minus this very small parameter h times this matrix of operators times this other matrix of simpler operators. They're all zeros off diagonal here, acting on our vector of functions. And it's equal to some right-hand side, which is computed. Again, you need to be able to compute the solution operator to compute the right-hand side. We write it out as, in simple terms, I minus h a b u is f. And what we can show is the following beautiful fact. The norm of the operator ab is smaller than some constant times what's called the Lebesgue number of the nodes. The Lebesgue number of the nodes has to do with the behavior of these interpolating polynomials in between the nodes. And this is why you have to pick the sample points carefully. For this choice of sample points, that number grows like the logarithm of the number of sample points, so very, very slowly. You would probably never see a nu larger than about 100. So in fact, the norm of this operator is just some constant times a very moderate number. And here you see in the formula, it gets multiplied by a very small number h. And so we feel good about trying to solve that equation. We're solving an equation which is a small perturbation of the identity. So here are some example solutions. We solved the equation with, so we have alpha is 0.2, beta is 0.7. The selection term is s is minus 5. And here are two different methods. And you see They give virtually the same answer. We solve from time 0 to time 10. I mean, I can't really explain much about these, but if you look here, you'll see the differences between the two solutions are about 10 to the minus 15. So we're really getting the same answer using these two completely different methods. Okay, here's an example where we solved with two different population models. One, a constant population at a half, another population of 1 plus t over 2. And here we see the two solutions. One is with black dots, one is with red dots. They're similar, they're different though. And over here what we have is a plot of the coefficients of the eigenfunctions. And what we see is that by the time we get to n is 25, the size of the coefficient is like 10 to the minus 16. Now why do we feel good about that? Well, we feel good about that because it means that we are really imposing the regularity condition. Because if our coefficients are decaying at that rate, it means that this is a series representation of a very smooth function, which is what we expect to see. Okay, now in the remaining minute, I would like to say that we don't actually use that method to solve the problem of interest, because the problem of interest has this problem that it's singular at the x equal to 0 edge for all times. And so we have to work extremely hard to resolve that singularity. And so my extraordinary collaborator, Jon Wilkening, worked out a way, well, we worked out this way to use the resolvent kernel, the inverse, like 1 minus h L non naught 0 inverse, to use the resolvent as a way to write the exponential as a function of the resolvent. And you think, oh, well, that's okay, because you can write down a formula for the resolvent. It's just a product of hypergeometric functions, which are slightly exotic, but perfectly okay. Okay, here's what happened. We wanted to take h small enough to get a lot of digits. And what Jon discovered was, for h of that size, the dynamic range of the resolvent kernel is 1 to 10 to the 13,000. So you have to ask yourself, how can you do a computation of a function that varies over a range of 1 to 10 to the 13,000? He figured out how to do it. Okay, he works with the logarithm of the solution. He uses what's called the Riccati equation, which is an equation satisfied by the logarithm. It becomes a much more manageable problem. At some point, you actually have to exponentiate the logarithm. So that's a tricky thing, but he figured out how to do that too. So hopefully, so the analysis is in a paper that we have not quite yet finished and put it on the archive. Hopefully it'll be there soon. Software hopefully will be freely available. I hope people will try to use this stuff and try to solve the Wright-Fisher, the Kimura diffusion equation again. And here I'll just close with a picture to show the solutions for one of the examples. So here's on the left-hand side, it's a linear graph from 0 to 1. And I want you to observe that the y-axis goes from 10 to the minus 25 to 10 to the 5. So you can see there's an incredible dynamic range in the solution. And on the right-hand side, what he did was he used a logarithmic axis on the x-axis as well. And so what you see, the solution is colored red to yellow to blue to green. That's the time. And what you see is that he's looking at the difference between the solution and the initial data. And here it starts at practically 0. The error is 10 to the minus 25 at time 0, more or less. And what you see here is that you get this sort of wave that moves up and to the right. And in the boundary layer, the maximum value assumed is 100,000 at time 0, at the last time step, which is 0.3. But you see that it's died down to back to 1 by the time the x variable gets to 0.01. So this gives you a sort of indication of how difficult a computation this really turned out to be. So then we sort of understood why Steve Evans simulated the stochastic process rather than solve the PDE. But we're hoping in the future people will solve the PDE, since now we've sort of figured out how to do it. Anyway, so that's all.